Yeah, good morning to everyone. Today my topic is on CNS tuberculosis. I'll be talking about whatever has changed in the last 5 years. I'll be discussing mainly based on the pathogenesis and how our approach to the therapy is still changing. So even now, after about 140 years of having discovered the bacteria, the pathogenesis is still poorly understood. So like how do they disseminate to the brain, and what regulates or dysregulates the host response, and what gives the bacteria added advantage to cause survival is still not known. So this is the first paper actually when Robert Koch actually showed to the world his famous postulates. So basically, he's the one who discovered this bacteria, and he's the father of bacteriology, as you already know. So coming to how it reaches the brain. So basically, the initial concept was the concept of rich is focus. So basically, somebody has a primary TB infection, aerosol droplets containing the bacilli are inhaled. They either seed hematogenously to the brain. But we've often seen that many patients whose lungs are totally clean, there's absolutely no lung lesions, also come with TB meningitis. So this hypothesis was not like holding good. Second thing they thought was whether it's a latent TB which had seeded during the active phase of pulmonary infection and then later reactivates in the CNS when the lung has already cleared. But again, the theory against it will be we don't, sometimes in those patients with severe TBM also, we don't see any scar or any evidence of previous infection in the lung. So again, how it disseminates to the brain and what causes, what activates the disease is still not clear. But once the bacteria is in the brain, it uses mechanisms. One, it enters the macrophages, and through macrophages, it's like a it's called a Trojan horse, where it piggybacks on the macrophages and enters into the blood-brain barrier, or it directly infects the microglial cell, and which causes secretion of cytokines, and that again causes inflammation. So then this leads to a breach in the blood-brain barrier. So once the blood-brain barrier is breached, it sits there, and all the macrophages which are infected attract more inflammatory cells, and that is what causes the formation of this Rich's focus. So usually the Rich's focus is somewhere in the subarachnoid space, most often found there, and further it enlarges and suddenly it ruptures. So the rupture of the Rich's focus is traditionally associated with development of clinical meningitis, and more foci leading to more exudates, hydrocephalus, raised ICP. If it's near the vessels, it causes ischemia, infarction. So this is the Rich focus model as just a summary of whatever I had said. The bacteria from the blood, and this is the brain, and this is the barrier. So uninfected cells, then it either enters through into the macrophage, the Trojan horse mechanism, where it enters, or it infects the microglial, causes the formation of the foci, and the foci ruptures and causes the clinical disease. So if you see here, this is just a picture, fluoroscopic picture, which is actually showing, you see this green ones here, which is showing the bacteria, which is inside the cells. And this is a endoscopic view of the third ventricle, where you see a lot of exudates, and this is again a picture of showing multiple tuberculomas, which are different. So how has our understanding changed? So basically, there are various ways in which all the manifestations occur. So the main problem, the first hit happens at the blood-brain barrier. So you have the macrophage, which is filled with the bacilli and the Trojan horse model, or the second one is the direct infection of the microglial cells. So inside the microglial cells, the next hit is the host cell recognition and internalization. So directly there is a neuronal infection. Subsequently, there is secretion of all these pro-inflammatory cytokines. The common ones are these, the TNF alpha, IFN gamma, IL-6. However, these are also found. Subsequently, because of the cytokines, there's inflammation. So the blood-brain not very tight, becomes permeable. So how does it become permeable? Because of these chemicals which are secreted. So ICAM, VEGF, VCAM, MMPs, and of course there is cellular influx of all these inflammatory cells. So this causes a very series of events. So there is endotoxin release, there is metabolic changes, there is infiltration of vessel wall, there are exudate formation, blood-brain barrier becomes leaky, and because of all this, you have varied manifestations in the form of hydrocephalus, edema, tuberculoma, abscess, arachnoiditis, etc. So there was another interesting concept where in the era where they did the Tuskegee trial, which you'd be familiar with, where they followed up these African people in the era of Nazi, where they infected the patients with syphilis, and they didn't treat them. They just wanted to observe them over the period of to see the natural history. So there were some unethical studies which were done on TB as well, where they found that about a very small proportion, in fact, clear the infection, even a CNS infection on their own. But after this ethics in research and all came into practice, all those papers were removed from the history. It shouldn't be quoted as what was there. It's mentioned in Robert Koch's biography, where later, almost about 50 to 60 years down the line, these type of studies were done. So now I'll be mainly focusing on the host immune response to TB infection. So this is one breakthrough paper which was published by an Indian called Lalita Ramakrishnan. So it came out in 2012 in the Cell. So basically, she said that this LTA4H, that is leukotriene A4 hydrolase, is the one which is in the pathway of arachidonic acid metabolism. So your arachidonic acid has leukotriene A4. So LTA4 hydrolase and 12 lipooxygenase. So it gets converted into leukotriene B4 and lipoxin A4. So this happens in normal, it's a normal metabolic pathway which is there in the brain. So in the whole body, basically, not just the brain. However, the leukotriene B4 is pro-inflammatory, helps in neutrophil and macrophage migration, whereas lipoxin is anti-inflammatory and stops neutrophil recruitment. So in the body, this pathway is in a steady balance. That is, the leukotriene B4 and the lipoxin A4 is equally balanced, so that inflammatory state. So whenever there's an infection, to take care of it, this balance tips over, and that's how the infection, in the attempt of infection getting controlled, you get inflammation. So what she studied was, she used a zebra fish. Zebra fish was like they say it's like a very transparent looking fish. So you can easily infect the bacteria on it, and you can actually see the granulomas very clearly. So you were able to, and she was able to delineate all the pathogenetic mechanisms, and she was also able to identify genotypes based on the levels of these enzymes. So based on that, she was able to say that there were two states. Either you have an LTA4H deficiency or you have an LTA4H excess, and both are bad. So if LTA4H is at the right level, it is good. So what happens when there is deficiency? There is LTA4 excess, and that causes TNF, because this will induce TNF. So if you have a deficiency, TNF is low. So you have an uncontrolled bacterial growth. So same way, when you have an excess, LTB4 is excess, TNF excess, that causes reduced intramacrophage bacterial growth and macrophage necrosis. So both were harmful. So based on this, they found 3 genotypes. They extrapolated this. They used Mycobacterium marinum for zebrafish model. However, in humans, they tried to extrapolate the same thing, and they saw in TB patients, they were able to find out 3 specific genotypes. One is CC, CT, TT, which exactly correlated to these LTA4H low state, optimal state, and high state in the zebrafish. And they took it to the next level, and they assessed how severe is the TB in these 3 genotypes. And they found that the TB severity is actually as you go to the TT and the CC, the TB severity was higher. And in the CT, the TB severity was lower. And they thought, okay, if we can tweak this inflammatory pathway, we give anti-inflammatory drugs, what happens? So then they found that if you give anti-inflammatory drugs to this group, they did well. And if they give it to the CC group, they actually did bad. So it's not a uniform response. Again, anti-inflammatory therapy also responded differently between the three groups. So with that, Thwaites is the major person with all the TB related research. So they looked at how does this genotype, and they also took HIV into consideration, and they looked at how intracerebral inflammation and survival among patients with TB meningitis, and they found that at all times, this is a Kaplan-Meier curve. So if you say A is for those without HIV, B is for those with HIV, and C with again looking at the case fatality rates between both the groups. So if you see, the red line is TT. So the TT people had a higher chance of survival at the end of 270 days, and the lowest survival was associated with the CC group, which was again confirmed in the like what was seen in the zebrafish model was confirmed in the human subjects. Another interesting thing, they also found that in those patients who are not HIV positive, that is immunocompetent patients, this LTFRH genotype correlated with even the cytokine expression in the brain. So for us, higher the cytokine, higher the inflammation. So in the cytokines, they found that the low concentrations of each cytokine in patients with CC, and they found intermediate concentrations in those with CT and higher concentrations in those with TT. So if you say, so the traditional theory was more inflammation, more damage. But this study refuted that whole hypothesis, because you're seeing TT who did well actually had lower inflammation but higher cytokine concentration. So there was some discrepancy here. So what they found was, so based on this, they decided that, so if you have a pre-treatment CSF cytokine concentrations, if increased, it correlated with a more severe disease. But in HIV uninfected patients, death was strongly associated with an attenuated inflammatory response. So if you have a lower cytokine concentration, you tend to do bad. Higher cytokine concentration, you actually had a severe inflammation, but you tend to do good. So again, this is a question mark. Why this paradox is happening in TB, we do not know. Other important thing is this hyperinflammatory TT genotype. This is the group when we hit them hard with steroids, they do very well. So because the steroids again takes care of the inflammation, though they are the hyperinflammatory state. But on the other hand, if you have a CC genotype and you give them steroids, already they have a attenuated response. On top of that, you suppress more. The outcomes are not good. So there were a series of good quality trials which have now confirmed that we have to select patients for steroid based on the LT4H genotype and HIV. But however, it's not yet like formally come out as a treatment guidelines, because everybody cannot employ this molecular method even in resource limited setups. However, they are also working on getting it like a card, like a point of care diagnostic kit, where you just take it, you just have to have one blood sample where you just put it as a like a ELISA, like a blot, and you get the result. But that's the trial. I mean, the studies are still underway.
It's only for TBM.
This can be used. That again, most studies, this is best studied in TBM, but they're using it for other organs as well, like pulmonary and.
Other organs we don't use it.
They are giving it for lung now. And disseminated TB also they are studying, but it's not in the recommendations yet. Like those people who have abscesses, like those type of patients, it's being studied, but not.
No, we are giving LTA4 as a service.
Yes.
We developed an easier method. We are trying to publish it. Like she said at that point of care, we can't reach that stage. But in molecular method itself, the older method which they use the related PCR. which is very expensive to have in smaller lab. Most of the time, these kind of tests you do in a small lab. We developed our own PCR and we have validated. And we are giving it as a service also.
So how long does it take, sir? The turnover, like if you give us
Our time is usually 1 week, but if you want us, we can use the result in 3 days.
2 days.
Sample which is
Blood sample.
it's a genome. It's a promoter genotype, so it is available in here.
Yes. So
See, one more thing, when they said that in CC and TB, the disease was much more severe. That is only TB meningitis. That's not.
yes, yes.
So severe in what sense?
Meaning they
Was there more
they actually looked at the MRS score. So basically, the severity for all Thwaites trials is the MRS grading.
This is the clinical trial.
Yes.
They adjusted for all the treatment and pre-steroid and all. Things like cytokines in the CSF.
Yes.
So many things that are contributing to it. And we have to be very dynamic.
Yes.
So many things like time in which the current state of the seizures.
Yes.
Duty is just a promoter. That's not in the coding, but still it is influencing. That is serious.
So which is better? Is it better to have a controlled inflammation, or you just do a immunosuppression? So again, if you look at this, so whatever I had said before, so this is the eicosanoid pathways. You have LT leukotriene B4 and you have a prostaglandin E2. So they are synthesized from the arachidonic acid. So at this level, you can stop the biosynthesis by inhibiting the COX and the LOX. These are the enzymes. So if you inhibit, this is the key to push the cellular events and polymorphonuclear recruitment. So what happens here is the more as the time goes, first you have the injurious stimuli. First is leukocyte infiltration. As it goes up, the edema peaks. After this initial phase, the polymorphonuclear cells, that is the neutrophils, get recruited into the picture. So looking at this pathway, they identified normally what all can block this pathway, which can help TBM. So first thing is steroids, which uniformly block it. Other thing is prostaglandins and synthetic analogs, NSAIDs, COX2 inhibitors, LOX inhibitors, leukotriene antagonists. But the side effects are, it's more, I mean, we just want the inflammation. If you immunosuppress again, it might cause more problem, because if you have live bacilli there, you completely wipe off the cells, you'll have uncontrolled bacterial growth. So that has to be balanced. So now based on that, they looked at aspirin, whether very high dose aspirin can help in TB meningitis when given early into the disease. So aspirin causes dose-dependent inhibition of this thromboxane A2, and they upregulate pro-resolving CSF protectins. This is body's natural mechanism to heal some injury. So they found that the same Thwaites group, they looked at 120 Vietnamese individuals with higher severity TB, MRS 2 and 3. So they found that there was a potential reduction in new infarcts and deaths by day 60 of treatment in microbiologically confirmed TBM patients. So they've actually, but the dose they used was quite high. They actually used 1,000 milligrams aspirin. But though they conclude like that, if you actually see the p-values are not like really significant between the groups. So this study, only thing is they found that in the aspirin 81 milligram group, 14.8 per 1,000 milligram, 10.7. And that was statistically significant. The placebo group, it was 34%, but it's not uniformly across the groups. So now that's then the third concept. If you have an inflammation, what does the body try to do? So the body tried to resolve with the help of two major molecules. One is resolvins, another is protectins. So these are actually called as SPMs. They are specialized pro-resolving mediator. That is, they push the body to auto heal itself. So they wanted to explore this whether this lipid, these are all lipids actually. If you turn the lipid switch on, whether it will stop the inflammation and promote healing. So they should find a way where you can potentiate this pathway, help in biosynthesis of lipoxins and maricins. So based on that, they found that so basically these protectins and maricins are all, as I told you, they are SPMs, they are specialized pro-resolving mediators. There are multiple complex names. They're all large lipid molecules which are there. So whenever there is an injurious stimuli, body naturally secretes it, but because the inflammation is too overboard, it cannot keep in pace with that. So once the inflammation is controlled, the amount of these SPMs which are secreted overdrive the to heal. So this can be good or this can be bad. Good in the sense that inflammation is getting controlled. Bad in the sense that healing causes fibrosis, adhesions, exudates, all those things happen because of them. So which indirectly leads to all the long-term like hydrocephalus and all those problems.
Very long treatment.
Yes, sir. Yes, sir. They're all lipids, sir. So again, after the neutrophils, the monocytes, macrophages, then your body tries to return to homeostasis governed by these SPMs. So now what they looked at is what is happening to these SPMs, these body's natural mechanism in healing in patients with TB, and does it actually link to disease severity? So this again the Thwaites group, where they looked at in MRC1, if you see, they have higher levels of this SPMs. Whereas when you have a higher severity, it has a lower level of SPMs. Again, why this paradox? We don't know, because we say that when you have a higher severity, your body's inflammation is more, and your previous pathway wise, this is very active, so this also should be more. But we found a different response where the healing mechanism itself is getting suppressed in higher severity. And similarly, the eicosanoids, which actually promotes further destruction, is getting potentiated when you have a higher severity. So again, what does that translate to? So if you have a survivor, they have a higher SPM, and a non-survivor had a lower SPM. So this again is mediated by some genotype like LTF4H, which has not yet been figured out. So the work is in process. So this will be like, if you find out what in the gene causes somebody to produce less SPMs, maybe that genotype have to be targeted more aggressively in the beginning itself. But what else can manipulate this lipid biosynthesis pathway is again a new question which is to be answered. So what are the bedside clues to predict mortality? So previously we know that somebody who had a higher protein, higher cells, lower CSF lactate, lower CSF glucose, all these patients have worser outcomes, and again your severity defines outcomes. So this again paper on 608 patients in Indonesia, they looked at all the factors from admission to discharge. So they found these four significantly affecting survival. So body temperature, of course, if you're febrile at admission, CSF to blood ratio, if it is lower, it predicts lower survival. And if you discard these two, this is of great interest, because from here many research started branching out. They found that higher CSF neutrophils, like more than 50 at admission, predicted a poor survival later. So something to do with this neutrophil response is causing bad outcomes. So they started to discover what are the neutrophil pathways which are already being described in great detail in rheumatoid arthritis and other SLE and all those things. So again, work is going on regarding the neutrophil pathways. Then next, coming to an area which is totally new for infection point of view, this is called as metabolomics, where previously lactate has been greatly studied because we blame it as a culprit of mitochondrial dysfunction. So now they found out this path. This is a serotonin and kynurenine pathway for which tryptophan is the backbone. So mycobacterium is very intelligent, I would say, because the bacteria requires tryptophan to survive. So initially it derived the tryptophan from human body. Then later we found out drugs which can drop the tryptophan. So what it did was it started auto-synthesizing tryptophan. So it has an inbuilt mechanism where it synthesizes tryptophan and derives energy out of it. So later they started identifying some chemicals which will interrupt the tryptophan pathway in the mycobacteria, but all these are animal studies and they found that the mycobacteria died. But all those, if you put it in human studies, they couldn't get the same result. But this pathway is again very interesting, where this MTB, when it activates inflammation, it causes cytokines, and these cytokines can trigger this kynurenine pathway. This kynurenine pathway is happening in the astrocytes. So once the pathway is activated, astrocytes and microglia produce kynurenic acid and quinolinic acid, where quinolinic acid is supposed to be the neurotoxic one, which causes excitotoxicity and increases the oxidative stress. So again, this is going to the genotype, where who has a higher risk of having this activation of serotonin and activation of kynurenine, and what role does tryptophan have to play. So they again looked at this study, see at cerebral tryptophan metabolism and outcomes. This is just an observational cohort, and they actually found that if you see this graph, this is glucose and this is tryptophan. So basically in the non-survivors, this is tryptophan. This is, yes, sir, this is CSF, sir. So if you see the tryptophan levels in survivors, it is low. In non-survivors, it is very high. So somebody who has a tryptophan level which is higher in the CSF are having a higher chance of non-survival. So how does this translate to clinical practice? We don't know, but it's just opened out a new way of, because it's tryptophan can be easily targeted by different drugs, but we don't have any further data, and again studies are going on. So last thing is the most often what we deal with, the paradoxical reaction. So basically we know that even after starting ATT, some patients worsen. They develop signs and symptoms of TB, or even go down after starting ATT. And these are called paradoxical reactions. So basically what they say is, when there is a high TB bacteria load, the inflammation goes up, then the bacteria dies. Then what happens? Inflammation still remains up. That causes more damage. That was the initial theory. But this is what was proven from the lungs. Basically, MTB goes, it accumulates in the macrophages, and it goes into the toll-like receptors. And there's something called as an inflammasome, which gets activated. So this will recruit new. Instead of it clearing and making things better, it causes all the bacteria and the macrophages to aggregate in one place, and that becomes a paradox. Basically, most often CNS paradox, what we see is tuberculosis. So the same thing happens in the lung. So somebody who has just nodules come back later with conglomerate big abscesses, or they have bronchiectasis and they have hemoptysis. So that is basically, again, the neutrophils are of interest here. So now they have found out that this is the next level, where beyond metabolomics, they did something called as transcriptomics. So they took the blood samples from the patients at admission, after starting ATT, and after they develop a paradoxical reaction. And they found that what was different in the blood transcriptomics, or the blood cells from the admission to this. So what they found was, all the iris was associated with a marked neutrophil dependent inflammatory activation. But this signature actually was traceable in the blood weeks before they come down with paradoxical reaction. So that was the most interesting thing. So actually, if we can find out some neutrophil specific marker, which we can identify in the blood, which correlates with the TB iris, we can use it as a biomarker to predict might develop iris, either taper the steroids slower, or we should think of some other strategy, because you can predict these patients will develop iris. So summarizing whatever I've said so far, so there is a immunodeficient milieu, there's an inflammatory milieu, and there's an optimal milieu. So if you have an as your this is a parabola of damage response framework. So if your host damage and bacterial growth increases, you get a immunodeficient. On the other side, you have a immunosuppression, you have hypoactive interferon gamma and CD4 T cells, you have macrophage necrosis from uncontrolled bacterial growth and high bacterial growth. That is when you have an immunodeficient state. But when you have an inflammatory state, you have high cytokines, you have hyperactive CD4 cells, you have macrophage necrosis, your bacterial growth is reduced, but later in the infection, it again comes up. Why? Because your neutrophils are coming and they're trying to cordon off all the bacteria. So in that time, you develop a worsening reaction. So there's somewhere in between is the optimal milieu where everything is in control. So most often we see this, but in paradoxical iris, we see this. In HIV malnutrition, we see this.
Sorry, this is the corticosteroids. This is the aspirin I already mentioned.
India also, I think, 2 years before UK Mishra group and one paper on aspirin.
Yes.
It's mostly for endarteritis.
Yes.
They have also demonstrated some benefit.
Yes.
That is 150 milligram.
Yes. But this is a high dose early in the disease course. This is to regulate the inflammation, basically. But again, we don't know safety factor also needs to be considered. After phase 3, we might have an idea.
Phase 2 would have said about safety.
Yeah, 1,000.
Meninges. So spine image showed a spondylodiscitis, and he had a large psoas abscess on the right side. Subsequently, in the what he was started on ATT steroids, he developed worsening hydrocephalus requiring a EVD. And he also underwent a pigtail insertion, which showed a PCR positive, but he had INH monoresistance. So CSF from the EVD showed 4 cells, low glucose, low protein, but expert was negative. he improved with EVD and this ATT steroids. He went back to Ranchi. 1 month later, he redeveloped worsening gait dysfunction and dysarthria. And clinical examination at that point of time showed worsening power, left upper and lower limb, and he also had some cerebellar signs. So they thought it could be some paradoxical reaction, or the VP shunt was not functioning, or endarteritis. So this is what the imaging showed when he came back after 1 month of ATT steroids, like multiple conglomerate tuberculomas and possibly one big abscess which is evolved. So they labeled him paradoxical TB reaction. They started him on infliximab therapy. So they gave monthly doses, 600 milligram. So he received 3 doses. So this is at last follow-up when everything has completely cleared. There's some just residual gliosis. Contrast. Contrast is also normal. I mean, I don't have time to. So he's ambulant with minimal support. He started walking. So, and they have about 22 or 23 patients who are almost went to the. I mean, almost were like almost like a mortality, and they pulled them back with infliximab. So these are some future research questions for anybody who's interested to do something here. So we don't know what is the mechanism for infarction, tuberculoma, and hydrocephalus. So previously was thought all basal exudates, all vessel, but what is actually causing this, nobody has still known. So how is the cerebral inflammation controlled by corticosteroids? That again, nobody knows. And what causes brain injury and relation to neuroinflammation? So some other research questions this is the first question which I am actually interested in. Can blood biomarkers be clinically used to predict development of paradoxical reaction? So we need to look at the neutrophil pathways, and we need some transcriptomic models to ascertain, confirm the hypothesis and find out whether, and based on the previous study, the most interesting thing is the blood signatures change weeks before. So if we can pick up those individuals, it can actually affect management significantly. These are other research questions. How can raised ICP be detected and monitored in low research setting? Should patients with brain abscess and CNS TB manage differently? And what is the best treatment regime for managing iris or paradoxical reaction? So all these questions we still don't have answers to. Thank you.
Very nice.
Thank you, sir.